Everybody, Ethan here. Today I'm going to talk about fatty acid beta oxidation. When I learned this in class, I'm going to be honest, it actually reminded me of something that happened to me when I was a kid. And before I explain that, I want to describe who I sort of was as a kid. And the truth is, I was a very self-assured little boy, irrationally confident. I think the hands on the hips in this picture really sort of encapsulate the vibe I had as a kid. And it really was one of ridiculous, irrational confidence. And fortunately, what happened throughout my life between then and now is simply put, I was humbled. That engendered a very necessary humility. And one of these humbling moments had to do with something that's a little gross, but I'm just going to explain it to here. It has to do with my discovery, my breakthrough of learning I could swallow my own saliva. And when I realized this as a young kid, I thought I had made a breakthrough of enormous proportions. Let me map out why. Okay, so in my irrationally confident young brain, this is how I sort of thought of how my discovery mattered. One, I got thirsty. Everyone gets thirsty, right? But I had made this discovery that I could swallow my own saliva. And by swallowing my own saliva, I could quench my thirst because, according to mom, saliva is made mostly of water. I can just, swallow my own saliva instead of drinking water. So with this sort of path in mind, if I tell others of this miraculous discovery, I could solve global water shortages and perhaps go down as one of the greatest scientists who ever lived. So again, this was the fantastical mind of young Ethan. What happened after this? Well, simply put, I remember thinking, oh my gosh, people who were thirsty could really benefit from this miraculous breakthrough if only they knew. So I decided to tell the first person that came to mind, which of course was my mom. I told her, Mom, what about swallowing your own saliva, solving thirst? Now you never have to drink water again. And she was like, okay, Ethan, why don't you try that for a day? Just try doing it for one day. And sure enough, after a few hours of being a hyperactive little kid running around, my saliva did not quench my thirst. If anything, my mouth became dry. There was nothing for me to swallow. And then unfortunately, I realized in a very humbling moment that my dreams of being a Nobel Prize-winning scientist for discovering the ability to swallow saliva were simply not going to happen. So what is the point of this ridiculous story? The point is to say that these failed attempts to hydrate myself with my own saliva indicate a pretty central truth that it also applies to energy metabolism. And that central truth is that our bodies cannot run without outside inputs. Everything's got to come from somewhere. So when we're talking about energy metabolism, I think glucose and, TCA, all this stuff gets all the credit. It gets all the flash for powering our cells. But the truth is this doesn't last long. It only lasts so long, and we can survive without eating for days, weeks, maybe even months. So what explains this? It's not some kind of magic loop. It's not like me trying to swallow my own saliva and thinking that'll, hydrate me forever. No, the truth is the energy got to come from somewhere, and it comes from what in my eyes is the unheralded hero of metabolism. That's right, fatty acid beta oxidation. So this PowerPoint or this lecture is going to focus more on the beta oxidation process itself. So there's a lot of prep points. I'm not really going into detail, but I'll cover them really quickly right here. So during these times of high energy demand, fatty acids enter the bloodstream, and usually that's coming from sort of the lipolysis of triglycerides in adipose tissues. So once you have these fatty acids in the bloodstream, they're uptaken into cells via fatty acid binding protein. And throughout all this process where beta oxidation is happening, you have to make sure you have enough oxygen that supplies there, and you have a waiting pool of these electron carriers, NAD plus and FAD, to get reduced. So that's got to be set up. And now you have all this set. And I'm going to explain why that last thing is important. But in short, we're going to start this story with fatty acids now in the cytosol. Okay. Once in the cytosol, they have to get activated by acetyl CoA. This reaction is two-step, and it's catalyzed by acyl-CoA synthetase. That's the enzyme that works in both of the steps. Before we even talk about the reaction, I want to sort of talk about what molecule we're dealing with. It's a fatty acid. Specifically, it's an acid, right? Fatty acids are acids, specifically carboxylic acids. And this R here, imagine it's a long carbon chain, right? So what happens this first step? You add ATP to the fatty acid. This reaction is catalyzed to create this adenylate, AMP intermediate, plus sort of PPI. What's PPI? It's pyrophosphate, if you were wondering. So now with this adenylate intermediate, you again, with acyl-CoA synthetase, add CoA to this, and you sort of substitute the AMP on this carbonyl group for a CoA. And what is this product? That's right, it's called a fatty acyl-CoA. The fatty is the R group, the acyl-CoA is this sort of functional group here. So that sort of makes sense. You're going from fatty acid to fatty acyl-CoA. One small detail I think may be relevant that I want to talk about is that this actually consumes 2 ATP equivalents. And I'm thinking, Ethan, why? You use only 1 ATP in this reaction. The truth is because you're converting ATP not to ADP, but to AMP. Remember, ATP is an enzyme triphosphate. It has 3 phosphates, 1, 2, 3. But the money-making bonds, the high-energy usable bonds in this molecule are these 2 here. They're called phosphoric anhydride bonds. These bonds are what are broken when you dephosphorylate ATP, and that energy, when you break the bond, is harnessed by the cell to power whatever it wants to power. Usually, cellular processes only break one of these bonds, turning ATP to ADP. But our specific activation here, what we're doing to make this fatty acyl-CoA, is turning ATP into AMP, which means we're breaking both of these high-energy bonds, okay? And that is therefore like double the cost of a normal ATP equivalent, because normally you just turn ATP to ADP, and then regenerating that to regenerate ATP, you just have to do 1 phosphorylation. But when you create AMP by dephosphorylating ATP twice, you have to convert AMP to ADP and then ATP, which is, of course, more energetically costly than ADP to ATP. So that raises the question then, if this is so energetically costly, why do we actually sort of activate this fatty acid? There's a number of reasons, but as you'll see, one of the main reasons is that CoA, which is this right here, is a necessary part of the transfer into mitochondria, the fatty acid to enter the mitochondria. And you also create acyl-CoA as an end product beta oxidation. And you can't create acyl-CoA without a CoA coming from somewhere. So we have this fatty acyl-CoA now. That's enough of all these asides. You have an acyl-CoA in the cytosol. What's next? Well, fatty acid beta oxidation occurs in the mitochondria, which means you have to get this fatty acyl-CoA into the mitochondria, which is a bit of a process. What's coming is a complicated Menzi diagram. I'm warning you, it's messy, but don't worry, I'm going to break it down. So this is the process of transferring a fatty acyl-CoA into the matrix. And as this diagram very incredibly neatly shows us, your fatty acyl-CoA gets converted into a fatty acyl-carnitine, which can then shuttle through these both membranes into the matrix, where it is then converted back into fatty acyl-CoA. But let's go step by step to really understand this. Step 1 is CPT-1, the CPT-1 enzyme, which is sort of in this outer membrane, catalyzing the substitution of CoA for carnitine. This is part of why activation with CoA is important, because CoA is a good leaving group, right? It's a bit of organic chemistry here, a good leaving group for the substitution reactions, acyl substitution. Side note, but an important one I want to talk about, CPT-1 enzyme here, it's inhibited by malonyl-CoA, which is a key intermediate in fatty acid synthesis, which is, again, the opposite of beta oxidation when we're breaking down fatty acids. And if you really think about it, it makes sense. If you're making fatty acids via fatty acid synthesis, you do not want to break them down at the same time. What's the point in that? It's like you make something, you break something. There's no point in it. What a waste of energy and effort, right? So by having malonyl-CoA, a fatty acid synthesis intermediate, inhibit CPT-1, which is a key step in fatty acid beta oxidation, you stop the creation of fatty acid acyl-carnitine. Therefore, these can't enter the mitochondria to get broken down by beta oxidation. And therefore, when you are creating fatty acid biosynthesis, you're also inhibiting beta oxidation, so you don't have 2 opposing processes running simultaneously. So that's an aside. Let's get back to what we're talking about here, which is transferring this into the mitochondria, this fatty acyl-CoA. So again, step 1, we convert via CPT-1 into fatty acyl-carnitine. What happens next? Well, this fatty acyl-carnitine can enter intermembrane space and then get exchanged or traded with carnitine to enter the matrix. So this fatty acyl-carnitine is now in the matrix. This fatty acyl-carnitine can join CoA in another substitution reaction, catalyzed by CPT-2 to create a fatty acyl-CoA as well as carnitine. Again, this diagram is not well drawn, but it's not like the carnitine disappears. It's just a substitution again. It's a bit messed up. I want to fit everything in, but again, substitution. You do liberate that carnitine, and then that carnitine can sort of return back out to the cytosol to do this process again. So in a nutshell, what are you doing? You're basically turning fatty acyl-CoA to a fatty acyl-carnitine and then turning it back into a fatty acyl-CoA. The reason why you're doing all this is just so that you can get it in to the mitochondria. Okay, so that was a lot of steps. So what is this, the end product of this whole process here? Now you have a fatty acyl-CoA in the matrix, which is right where it needs to be, because now the stage is set to actually do the topic of this presentation, which is fatty acid beta oxidation. Before I go into the nuts and bolts, I want to provide a high-level overview. I think it helps illustrate and elucidate the concepts a little bit better. What is beta oxidation? It's oxidation at the beta carbon. What's oxidation? Remember, Leo says Gur, that's what I learned in high school chemistry. Oxidation means losing electrons. So you're making more bonds to more electronegative atoms. This beta carbon is making more bonds to more electronegative atoms. And the goal here is to cleave these long chain fatty acids into acyl-CoA, which can be used in the TCA cycle, and also generate NADH and FADH2. If you recall from the beginning, this is during a high energy demand state. So generating acyl-CoA to run TCA and generating these NADH and FADH2 is all good, because that's all stuff that helps you run TCA, helps you run ETC, which is going to generate energy. So let's take a closer look now at our molecule of interest, which is our fatty acyl-CoA. When I said we're oxidizing the beta carbon, that means that it's beta to the carbonyl, which means this carbon here. And remember, this X here refers to that long carbon chain. So this is the sort of substrate we're dealing with. And again, when we go back to this, I just want to remind you all that the goal here is to create acetyl-CoA, which can then generate energy. And again, this is good because we need energy during times when we're running beta oxidation. And I want to be very clear. Glucose metabolism, right? Glucose also makes acetyl-CoA. And you might be thinking, what's the point? Does this seem a little redundant that we're doing all this beta oxidation, all this work to make acetyl-CoA with glucose do the same thing? And it's true. Glucose does do this. And it is fast and flashy. I like to think of glucose as like a Ferrari, right? It's flashy, it's big, everyone thinks about it, and it's impossible to ignore. But what happens when the Ferrari runs out of gas or it is in the shop? What do you turn to? That's right. You turn to your trusty Toyota Prius. You turn to beta oxidation, which is the Toyota Prius of cellular metabolism, because yes, it's not as fast. You might have to wait to cleave your fatty acids from your triglycerides and do tons of steps. But when you need energy and glucose isn't there, this fatty acid beta oxidation is there. It's reliable. So dumb analogies aside, the point is that this fatty acid beta oxidation seems redundant in that it's also making acetyl-CoA the same way that glycolysis sort of and the glucose metabolism cycle would. But the point is that during high energy demand states when glucose metabolism isn't there, when perhaps you're also running gluconeogenesis and you need power to do that, that's where this fatty acid beta oxidation steps in. So let's, again, analogies aside, I want to take another step back before stepping in. When I think of this in a big picture, again, the goal is to cleave a 2-carbon acetyl-CoA from a fatty acid. So stepwise, what you're doing is you're oxidizing this beta carbon. You're making a double carbon bond, then a single bond to oxygen, then a double bond to oxygen, and then therefore these steps allow you to reduce electron carriers. This oxidation allows you to create or reduce electron carriers that can be used in the electron transport chain. So again, it's important. I like to think this way because by focusing on the beta carbon, sort of what I was saying, okay, look, the single bond, now it's a double bond to carbon, now it's a bond to OH, now it's a double bond to O, sort of illustrates how you are doing this stepwise oxidation, getting oxidized, which allows you to reduce electron carriers and set up a good leaving group for cleavage. So again, enough big picture stuff. Maybe that didn't help you. Let's go step by step. The first step in beta oxidation is oxidizing that beta carbon to form a double bond. I like to think of the chemistry reaction. And another way you can think about it, instead of just thinking of the beta carbon losing electrons, because now it's forming another bond to a more electronegative atom, instead of hydrogen, it's now bonded to carbon. Another way I like to think about it is you are creating an FADH2 from this, or I should say you're reducing FAD to FADH2. Where does those H's have to come from? Oh, that's right. It has to come from the beta carbon that was once, had these two free hydrogens, but now because it has a double bond to carbon, the hydrogen from here and the hydrogen from the alpha carbon end up going to FAD. So again, just another way of thinking about it, right? You have to put the hydrogens, when you create this double bond somewhere, they go to FAD. So this reduction of electron carrier is great because now I can go with ETC to make energy. But again, this is just the first oxidation of the beta carbon. The second step is another oxidation of the beta carbon. Now you're hydrating, you're adding water via this enzyme here. And now this beta carbon is bonded to a very electronegative atom, which is oxygen. So now you have this intermediate. and we're going to oxidize it a step further. So now we're using this enzyme, 3-hydroxylacyl-CoA dehydrogenase. We're going to turn this OH into another carbonyl. So now this carbon has 2 bonds to alternative atom, in this case oxygen. And again, that H has to go somewhere from the OH. It's going to NAD. It's able to reduce NAD plus to NADH, which is great. Again, another electron carrier that goes to the electron transport chain. But now you have this sort of double carbonyl intermediate, which actually is perfect. It's because it can be substituted on the same way that, we added carnitine to the acyl-CoA and substituted there. You can also substitute onto this beta carbon. It is a carbonyl, and that's what actually happens in the last step of beta oxidation. Via this ketoacyl-CoA thiolase, you sort of bring in CoA, and that CoA is attacking that beta carbon, the carbonyl, and is able to substitute and therefore kick off this 2-carbon atom, or not atom, 2-carbon molecule, which happens to be acetyl-CoA. So now this acetyl-CoA is now cleaved off, and it can now go to the TCA cycle to make more energy. And what's important to know is that this, of course, can be repeated because this X is more carbon chains. You can just do this whole process again and again and again and again. So let me sort of take a step back and review. What did we just cover? During high energy demand, fatty acids are released to the bloodstream and uptaken by FABP. Acyl-CoA synthetase converts fatty acid to fatty acyl-CoA. Fatty acyl-CoA moves in the matrix via reactions involving carnitine, catalyzed by CPT-1 and CPT-2. And lastly, the actual beta oxidation, these acyl-CoAs are oxidized stepwise, step by step, at their beta carbon to reduce electron carriers and cleave off acetyl-CoAs. So that's really important, right? Because this allows you to generate these electron carriers and acetyl-CoAs that can run the TCA cycle and electron transport chain in the absence of, a large amount of glucose. So instead of running glycolysis and stuff, you can just do this to sort of run those things as well. And this does create energy, which again, because you're in the fasted state, also helps you power gluconeogenesis, which is important because not all cells can do the beta oxidation. This mostly happens in the liver. So by able to generate all this energy, you can then power gluconeogenesis to create glucose so that other cells can just do the normal glucose metabolism cycle, glycolysis, TCA, electron transport chain. It's also important to note that what we talked about, this whole process, gets repeated again and again and again. So I did say that before, but I want to say it again. We're talking about this 4-step process here, but imagine if you have a long-chain fatty acid, you're going to eventually cleave that whole chain into tons of acetyl-CoAs, right? You're just going to repeat this again and again and again and again. And this, of course, only works if you have an even number of carbons, because acetyl-CoA has 2 carbons, can't make an acetyl-CoA out of 1 carbon. So if you have an odd number of carbons, it's going to take a different path. This video is already too long, so I'm not going to talk about that, but I'll post some links in the description if you want to see how beta oxidation happens in those odd chain fatty acids. But yeah, in short, that's beta oxidation. In my mind, it's the unheralded hero of energy metabolism that steps in when glucose metabolism isn't there to generate energy to do all the stuff that you need to do, which includes making more glucose for cells that can't do this unheralded heroic process. So yeah, that's beta oxidation. Here are my references. I appreciate you listening. Yeah, take care. Wish you the best.